Hi, and welcome to Chapter 10, screencast on muscles. As this says right here, I have updated this slideshow a couple of times because I just can't decide how best to present this material. So if you haven't downloaded the file from OneDrive using the link in week 10, get this file from the link so that you have the most up-to-date version, which I am using, which seems to change by the minute. At any rate, let's just dive in and see how I do with the slides I have. Oh, why am I not advancing? Here we go. All right, so first up, just know these functions. I tend not to ask too many questions about this. It's pretty obvious to most people what your skeletal muscle does. It moves you around. You also want to remember that you have smooth muscle and cardiac muscle, which moves things like blood through your veins, and then your smooth muscle controls blood flow and moves food through your digestive tract. So your muscle tissue overall does more than just move your body around. 2, posture, fine. 3, stabilizing joints. Just don't forget that muscle tone, that tension across all of your muscles or in all of your muscles that cross your joints, that is the major stabilizing factor for your synovial joints. And then your muscles also produce a lot of heat, which we tend to forget about, but it's actually a really important biological function and one of the things that separates us from lots of the animal world. So let's dive right into the more important stuff. The first thing we want to understand is the structure of a muscle, and we are going to start with an individual muscle fiber. So each one of these is one individual cell, which we call a fiber because it's long. They can be up to 30 centimeters long. So that's a foot or more. And you'll notice when you look at this fiber here, there's a nucleus here, a nucleus there, a nucleus there, and a nucleus there. So what your muscle fibers are is actually the product of the fusion of what are called myoblasts. So you have these immature cells that are going to become muscle cells that all line up next to each other, and they form this is what's called a myotube. But then the separations between them go away, and you're left with one long cell that still contains all of the original nuclei from the separate cells that all fuse to make this muscle fiber. So that's why you see all of the nuclei. And this is the first thing we want to understand. And that's why we call them multinucleate, because they're fusion cells. Then, so we start down here with a single cell. We're going to ignore the myofibril for now. We'll talk about what they are later. This is one individual cell. So this here is one of these. If you take a bunch of cells, this is supposed to be a cell, right, there's your nucleus as part of that cell, and you bundle a bunch of cells together, that is called a fascicle. And then what a muscle is multiple fascicles. So this right here is this fascicle here, and then you bundle together all of these other fascicles to make this organ called a muscle that has not only all of the muscle cells, but it has connective tissue and blood vessels and nerves all embedded in it. So then layered on top of this, we want to know the different layers of connective tissue that hold all of this together. So one of the things we want to remember about muscles is that almost literally every part of a muscle is attached to some other part of that muscle, so that mechanical forces are transmitted throughout the muscle. So you don't have to contract every cell, every fiber of a muscle to get that muscle to move as a unit. So we want to think of everything in a muscle being directly or indirectly connected to everything else. So if we start here, this is our individual muscle cell inside of a fascicle, and all of the little white space in there, that cream color, that's connective tissue called endomysium. It is made out of areolar connective tissue. Then surrounding the fascicle, bundling all of those individual fibers together, is perimysium. And then around the entire outside of the muscle, bundling the fascicles together, is epimysium. And epimysium is made out of dense, regular connective tissue. And you are going to see that it's the epimysium that actually forms the tendon as well. That's the part of the muscle that attaches to the bone. So your tendons are also made out of what is called dense, regular connective tissue. So now we're going to go back to this individual cell, which we call a muscle fiber, and look at some of the parts of it. So here's where we're going to talk about that myofibril we were talking about. So each muscle cell contains many of these long cylindrical protein-rich structures called myofibrils. And then if you look closely at a myofibril, what it is composed of are sarcomeres. So this here, which runs from this Z-disc to that Z-disc, this is one sarcomere. We will talk later about what makes up a sarcomere. For now, we're just going to mention them, and then we'll come back to it. So if you look at this one myofibril here, this is the middle of a sarcomere. Here's another sarcomere attached to it, and there's another sarcomere there. So this myofibril, much like this one right here, would run the entire length of the cell. And all of the myofibrils within this cell are lined up in register, which means the middle of one A band is on top of the middle of another A band, and that's why these stripes run down the whole thickness of the cell. So this is one cell thick here, many myofibrils running horizontally in this direction, but the striations go straight down because all of the myofibrils are lined up with one sarcomere in register underneath the next, underneath the next, underneath the next. And that's why you get these regular striations. So other parts of a muscle cell that we want to understand. One is we just need to introduce this new term called the sarcolemma. It is the cell membrane of a muscle cell. But because this is a highly specialized cell, and this membrane has some highly specialized proteins that you don't find in many other cells, we give the cell membrane a special name, and we call it the sarcolemma. There are places where the sarcolemma actually grows down and penetrates into the interior of the cell. These are called Ttubules. So this hole right here goes down into this tubule. This tubule will wind its way the whole way through the cell. So if you had, let me pick a color that I think is going to show up, a very dark purple. Right, if you had a T-tubule here, a little hole, it would wind its way through the whole cell. And I can't go around all of the, sarcomeres. But if you put a drop of water in the T-tubule here, it would come out down here. So they go the whole way through the cell from one end to the other. When it says continuous with the sarcolemma, that means the material that makes up the T-tubules is the same kind of phospholipid bilayer that makes up the cell membrane. And later on, we're going to talk about proteins in the sarcolemma that allow your muscles to respond to nerve impulses. Those same proteins are embedded the whole length of the T-tubules. And then the T-tubules are next to this darker mustardy looking yellow called the Sarcoplasmic Reticulum. This is a derivative of the endoplasmic reticulum. So it is a special adaptation of endoplasmic reticulum that you find in muscle cells, so it gets a special name. Its special function in muscle cells is to store calcium. This is one important thing to point out. Sort of this space right here, all of this space that's inside the cell, but not inside of the sarcoplasmic reticulum, this is all the actual cytoplasm, or what some videos you are going to watch might call sarcoplasm. And if you're inside of the sarcoplasmic reticulum, you are separate from the cytoplasm. So you want to think of the sarcoplasmic reticulum as like a storage closet. If there's a tool that you need to use or something that you want to work on your desk, you can't use it if it's in the storage closet. So it keeps the calcium separate from all of the parts of your myofibrils that are actually going to be completing your contraction. So now we come back to our myofibril, and maybe I should just switch things around and put myofibril at the end. I'll change it later, not for you guys. Where was I? So going back to the myofibril, now we're getting down into the nitty-gritty structure of a sarcomere. As we said, one myofibril runs the length of a cell, and it is a series of sarcomeres that are lined up end to end to end. This is the contractile unit of a muscle. It is the sarcomere that gets shorter, and because all the sarcomeres are attached to each other and to the outside of the cell, which is then attached to everything else, right? Because everything in a muscle is indirectly attached to everything else in a muscle. When all of the individual sarcomeres get shorter, the whole muscle gets shorter. So they extend from Z disc to Z disc. They have these purplish maroon thick filaments and these green thin filaments. We want to know what makes up all of these parts. Oh, I forgot. And then looking at this picture, right, each one of these dark bands that we call the striations, that's the A band here. That's the thick filaments that stain darker than where you just have the thin filaments and the Z lines. So looking then at a sarcomere, this is your Z line or Z disc. I am used to calling it the Z disc. This is where your thin filaments are anchored. So they cannot move without moving the Z disc, which is attached to the sarcomere next to it. Right? Again, everything is attached to something else. Then that thick filament has running down the middle what is called the M line. This is a protein called myomesin that connects all of the thick filaments to each other and prevents them from moving independently. So you have thick filaments all connected to each other in the middle, and ultimately connected to the cell membrane, and then the thick filaments connected to the thin filaments, which are connected to the Z line. Then let's look a little bit closer at our thin filament. Our thin filament actually contains 3 different proteins. The green balls that you see here, those are made out of actin. So one individual molecule of actin is a small globular protein, and then they stick to each other, one to the next, and form this long filament of actin. So this is what is called polymerized actin. Each actin molecule has a little spot on it that myosin likes to stick to. Myosin is the protein in the A band, or the thick filament, which I should have talked about first, but whatever, just remember that myosin binding site is there. And it is normally covered by this long filamentous protein here called tropomyosin. And then attached to the tropomyosin is troponin. So the way to keep tropomyosin and troponin straight is that the longer protein, tropomyosin, has the longer name, and the smaller protein, troponin, has the shorter name. So thin filaments composed of actin covered by tropomyosin and troponin, and then your thick filaments are composed of many, many, many molecules of myosin. So down here is one myosin protein. This is a protein that has what you would call quaternary structure because there are two golf club shaped polypeptides or amino acid strands that get twisted together to make the mature molecule. So here you see the tail with the heads pointed in this direction, and that is what this side here of this thick filament looks like. If we were to look at the other side of the thick filament, we would see all of the myosin heads extended in the other direction. So yeah, like some of them on this side are extended that way, and then on the other side they're extended that way. And you're going to see when movement happens later, these myosin heads actually move each toward the middle. And myosin then is the motor protein that actually generates the shortening of all of your sarcomeres during a contraction. So that then brings us to a contraction. What is a contraction? So if you look up here, there is a little bit of space in between our thick filament and our thin filament, meaning that our myosin is not attached to our actin. So they are not in contact with one another. When your muscles are going to perform a contraction, something happens which I haven't told you yet, which will cause myosin to be able to come into contact with these myosin binding sites here on the actin. Then myosin will be able to grab hold of the actin and pull it towards the M line. So you remember these myosin heads are pointed in that direction. These myosin heads are pointed in that direction. These are going to pull in this direction. These are going to pull in that direction. So as you can see here, right, the whole sarcomere, if I draw, whoops, I can't even draw a straight line today, or any other day. Right, the whole sarcomere shortens a little bit. The Z discs used to be out here, now they're pulled in and they get shortened a bit. So each sarcomere shortens when myosin pulls on actin and the two Z discs get pulled closer to each other. And because each sarcomere is part of a long filament of sarcomeres called a myofibril, all the myofibrils get shorter at the same time, and the whole muscle cell gets shorter at the same time, or the whole muscle fiber. So that is sort of the overview of how a muscle cell is put together and the super simple definition of a contraction. Then what we need to do is go back in and layer some real physiology on top of all of this. So this is what is called excitation-contraction coupling, and we're going to be on it for a little while. So what excitation is electrical excitation, which takes place where a nerve and the muscle cell come into contact with one another. This is going to ultimately cause myosin to grab onto actin and pull the actin towards the M line. There has to be something to connect the electrical activity that starts near this neuron with the contraction of myosin and actin. So the mechanism that couples these two things together is called excitation-contraction coupling. There are a couple of homework assignments that I'm going to have you do. And I'll just give you a heads up where the way I explain things is we go over all of the structures that you can see where the neuron and the muscle meet. That is called the neuromuscular junction. And we review all of the concepts and the vocabulary that you need to know associated with the neuromuscular junction. And then we do excitation-contraction coupling as one long continuous story. The video I'm going to have you guys watch, or the videos, breaks this up into two separate steps. So they talk about the function of the neuromuscular junction and how excitation works. And then in a separate video, they start with excitation and finish the contraction. So they split the information up a little bit differently than I do, but the information is pretty much still the same. So what we're talking about now, again, is the mechanism that couples electrical excitation, which starts on the surface of the muscle cell, with the contraction of myosin and actin. And you're going to see, like, spoiler alert, calcium is involved. So the way your muscles are controlled is by neurons that extend from your spinal cord. They are called motor neurons. So all of your motor neurons begin either in your brainstem or in the gray matter of your spinal cord and extend in nerves all the way through the periphery. And then they come into contact with individual muscle cells at what is called the neuromuscular junction. And I think I covered everything on that slide. Then if we zoom in on the neuromuscular junction, we get something that looks like this. So this, let me go back one. This long process right here, this is all one cell. It can be as long as your arm or leg. This part of the neuron that extends from your spinal cord to the muscle is called an axon. The end of the axon, that part right there, is called the axon terminal. It is where the axon ends or terminates. So inside your axon terminals are vesicles filled with a neurotransmitter called acetylcholine. So all of the motor neurons that control your skeletal muscle release acetylcholine. That is contained, again, within vesicles stored in the axon terminal that is going to be released by exocytosis. You will notice that these two cells do not come into direct physical contact with one another. There's a little gap in between them called the synaptic cleft. So these vesicles are going to dump neurotransmitter into the synaptic cleft. On the other side of the synaptic cleft is a special region of the sarcolemma where you see all of these folds. That is called the motor end plate. It gets a special name because it has a special function. What makes your motor end plate special is that it contains these proteins here called acetylcholine receptors. This is going to lead us to our next topic, which is ion channels. And we'll pick back up and talk about what kind of ion channel acetylcholine receptors are in a few minutes. But for now, we need to introduce this topic of ion channels, which you're already somewhat familiar with. So we saw channels when we were talking about the resting membrane potential, these potassium channels that are always open. Those are called leak channels. It's just a hole in the membrane that an ion or ions can pass through. Some channels can open in response to stimuli. Those are called gated channels. They have the ability to open and close. Generally speaking, all of your gated channels are closed most of the time, and then open under special circumstances. So if you have a voltage-gated channel, this is a channel that will open in response to a change in voltage or a change in the membrane potential. So over here we have an ion channel. You can see that it is plugged shut. Right here it is plugged shut. Right here it might be kind of small. The voltage is negative 70 millivolts. When you get to negative 50 millivolts, that change in potential, or that change in voltage, causes this channel to pop open, and now ions can pass through it. Over here we have what is called a ligand-gated ion channel, or a ligand-gated channel. So a ligand is anything that sticks to a receptor. So this here is a receptor, and how should I put it? Ligand is just has a functional definition. There is no structural description of what makes a ligand a ligand. It is defined as whatever sticks to and activates a receptor. So in this case, here's acetylcholine, which is going to stick to the receptor there, and then the channel or the receptor pops open, and now ions can flow through it. So you are going to see that this ligand-gated channel is what you have in your neuromuscular junction. They are acetylcholine channels, which we'll talk about in just a moment. Let's see, we're 26 minutes in. We should probably take a break before too long. I just want to review potential real quick because I said we would. You may remember that I said potential is a separation of charge across the membrane. So the resting membrane potential is the separation of charge that you find across a membrane at rest when it's not being stimulated by a neuron or some other type of stimulus. And it's normally somewhere around negative 70 millivolts. So every cell in your body has a resting membrane potential that is somewhat negative. And we measure that on the inside. So you may recall here we have a cell. Then let me grab a different color. Oh, that's too thin. Whatever. This is our sodium-potassium pump. And a pump. Does anybody listening recall which way the sodium is going to be pumped? You may recall sodium gets pumped out of the cell, which then means, let me pick a different color, nice green, potassium gets pumped into the cell. So you have a lot of sodium outside the cell, a lot of potassium inside the cell. And then let me just draw this real quick. Right, there are your potassium leak channels, which lets the potassium leak out. So here is some potassium leaking out of the cell, positive charges. So positively charged potassium leaks out of the cell. You have charges inside the cell, anions, oh, that's really thick, that want to follow the potassium out, but they can't because they can't pass through that channel. So you end up with the anions accumulated on the inside of the membrane and positive charges accumulated on the outside of the membrane. So you're negative inside compared to outside. That is your resting membrane potential, which is caused by the leakage of potassium down its concentration gradient, that gradient being set up by your sodium-potassium pump. So the same thing is happening in your muscles because every cell in your body has the sodium-potassium pump, and a resting membrane potential that is somewhat negative on the inside. Let's just set that aside for now. So in this state, where you have a potential across the membrane, negative on one side, positive on the other, we say that the membrane is polarized. So anytime there is some sort of separation of charge, a potential, it is polarized. If you move towards zero, that is called depolarization. So at zero, you are totally depolarized. If you move from negative 70, let's say, to negative 50 millivolts, we would still say that it is now somewhat depolarized. So anything closer to zero than your resting membrane potential is called depolarized. You can polarize it in the opposite direction, positive on the inside, negative on the outside, but we generally don't talk about that. I probably shouldn't have even put it there. Then if you go from depolarized back to your resting membrane potential, we call that repolarized. So you reestablish your resting membrane potential. This is why it's called the resting membrane potential, because the basic setup of the cell has this equipment that causes the resting membrane potential to happen. So any cell that is not being disturbed by some neurological or external stimulus is going to have that negative on the inside resting membrane potential. So as I said before, your cell is almost never at zero. There's usually some separation of charge, some potential across the membrane. So at rest, as I said, we call that the resting membrane potential. But there are lots of times when your membrane potential is going to change. Each time it changes, there's still a potential. So each different kind of change, depending on how or when the membrane potential changes, we give it a name, but it's still called a potential. So you're going to have action potentials. In AP2, we'll talk about receptor potentials and end plate potentials and postsynaptic potentials. These are just different circumstances under which the resting membrane potential changes, or the membrane potential becomes something other than the resting membrane potential. So we're going to see this word potential a lot to be used to describe different states of the voltage across the membrane. Then, actually I'm not going to use this blank thing yet. Let's go here. So back to our neuromuscular junction. I know it's been a long road. We're 33 minutes into this. So here we are again. We've got our axon terminal with its vesicles that contain acetylcholine, and then on our motor end plate, the part of the sarcolemma or muscle cell membrane right underneath that axon terminal, we have our acetylcholine receptors, which now over here we have labeled as ligand-gated sodium channels, which means they are channels that are going to respond to the presence of acetylcholine by popping open, and they primarily allow sodium to flow through them. Some potassium can flow through them as well, but much more sodium. So the overall effect of opening one of these acetylcholine receptors is for sodium to flow through. So we're seeing the effect of sodium flow on membrane potential. So let's go back to a cell. Oh, now our cell is green. That's fine. All right, it's negative on the inside. Then we're going to call this our acetylcholine receptor. At first it is closed, but then acetylcholine shows up. This is ACH. It sticks to the receptor. Now our receptor is going to pop open. Pretend this is the same receptor over here, only now it's open. Right, right? So our ligand, which is acetylcholine, caused our receptor, which is actually a channel, to pop open. Now sodium can flow wherever it wants to. Given that sodium is higher inside or outside, anybody? Outside the cell, let's see, let's make sodium green, right? You have a lot of sodium outside the cell, sodium is going to flow inside the cell. Now the inside of the membrane is going to start to look a little bit positive, or at least not as negative as it used to be. So you might go from -70, say, to -50. So we come back here, sodium is going to flow into the cell. That is awful penmanship, Dr. Iannacone. Into the cell. So does that mean the membrane is going to repolarize, depolarize, hyperpolarize? Can anybody guess? It is, right, depolarize. So if we go back here, just for fun, I like black when I'm drawing graphs, even though it doesn't really matter what color you are. So here's -70, right? That's our resting membrane potential. Let me just draw a straight line, it's easier. Then when acetylcholine shows up, sodium rushes in and the membrane slightly depolarizes. Let me just make that continuous. Then once this closes, the resting membrane potential is going to be reestablished. But you do get a brief depolarization in response to those sodium channels opening. That brief depolarization, let me skip ahead, is called the end plate potential. And that is where we are going to pick up in our next screencast. Because it has been almost 40 minutes, and that is a lot to digest. And I just want to take a break. But this is the first step in excitation-contraction coupling. This is sort of part one of a two-step excitation process.